Gresham College presents Whither to the Creeping Paralysis by Professor Chris Shaw.
It's a great pleasure to be here. It took me some time to find the place, but now that I'm here, it's good, and I'm really pleased to see so many people come out on this cold night to hear this talk. I'm a neurologist. I see people with motor neuron disease, and I have been working in this field for nearly 20 years. And it's a field in which has changed dramatically, and hopefully by the end of the evening I can persuade you that we are making progress and we are in a better position to discover a cure for this dreadful disease. So I thought I'd talk a little bit about what the disease is. This is a drawing by Gowers of a patient with motor neuron disease. We're going to talk about the diagnosis, how as clinicians we make the diagnosis, what are the symptoms and signs, what treatments can we offer. I'm also going to talk about the disease process itself, the pathology, what actually is happening in the brains and spinal cords of our patients, and in particular focus on the proteins that are accumulating there. And then going to talk about the genetics. When I started, I was working in the genetics department and the neurology department as a sort of hybrid. And I said I was interested in the genetics of motor neuron disease, and the geneticist said to me, "Oh, that's not genetic." So obviously I was in a tough spot. But subsequently, we have discovered a number of genes, and I'm going to talk about two genes in particular that were discovered in our laboratory, namely TDP43 and fused in sarcoma. You'll hear more about those later. So these are two things. One is we can obviously advise our patients who come from families in which motor neuron disease runs as a trait, and we can help them in terms of being tested. But we can also use these gene defects to study the disease process in cells and animals, and we can then hopefully target our therapies more effectively and also screen them to see what might work in our patients. And lastly, I'm going to finish with a personal message and talk about other people's experience and give you some information about that. So what I'm going to do is run through my talk. We'll have a period of questions, and at the end I'm going to show a video. And you'll understand at the end, after the video, that there probably wouldn't be many questions asked after that. It's quite a powerful video, and I should warn the sensitive in the room. It can be quite distressing. Anyway, this man, Charcot, was the first person who really clinched this as being a disease based on the degeneration of motor neurons. And he gave it a terrible name, I have to say, called amyotrophic lateral sclerosis. Okay, it's because as a pathologist, he was looking at the spinal cord, he saw that the motor neurons were degenerating, and he's drawn two motor neurons there. This one it has an inclusion protein accumulating in the cell outside the nucleus, and this one, which is obviously stuffed full of something proteinaceous. And he also showed that there was scarring in the lateral corticospinal tracts. So that's the lateral part, and the amyotrophic refers to the wasting of muscles. So most of the world actually calls this ALS. Now, Lord Brain, and that was his real name, Brain, and he was a neurologist, decided a much more sensible name was motor neuron disease, but of course it never really caught on. And in America, it's named after a famous baseball player called Lou Gehrig, who died of this disease. And so you'll hear it referred to as that. But the name I think is most evocative is the creeping paralysis. And this is a colloquial name that was written on death certificates through the Middle Ages right until the 18th and 19th century, which really does describe the nature of the disease in itself, and I think actually has much more power in terms of depicting that. Well, that is Lou Gehrig, and he was a very wonderful baseball player. He almost never missed a game. The year before he was diagnosed, however, his batting average dropped, which did indicate that there was some subtle motor problems, and he was dead within 18 months of diagnosis. This is Don Revie, who's a footballer, and there is some excitement about the number of footballers that have developed this disease. I'm not sure whether it's just because they're young fit men, but there is a theory amongst many of my colleagues that occupations in which there's a high degree of exertion predispose people to getting motor neuron disease, which is possible. And of course, this rogue, David Niven, died of this disease, and he has given his symbol, in his last days, the thumbs-up symbol, which the Motor Neuron Disease Association has kept as their emblem for their association. This is Jill Tweedy, the author and journalist who died of motor neuron disease, Stephen Hawking, who needs no description, and we believe that Mao Zedong died of this disease. Although, of course, I wasn't his physician, and I'm not sure we have really clear information on that, but certainly there were features that suggested that he had motor neuron disease near the end of his life. Well, where does it occur? In fact, it's a global problem. There are no communities that are free of motor neuron disease, but it has a very high incidence in two areas, one in the Key Peninsula in Japan and one in Guam. Now, the Guamanian one is slightly atypical in the sense that it's also associated with a form of Parkinson's disease and dementia, this trio of disorders. And sometimes they come in combination and sometimes they're separate. Interestingly, it had its peak in the 1950s and then has been decreasing over time. And there's pretty convincing evidence that there's an environmental component to that, perhaps cycad consumption. More men that are affected than women, a ratio of roughly 1.7 to 1. And it's a disease that comes on in the best years, shall we call them, in later life. But in fact, it does affect people in their 20s, and I've seen several teenagers with this condition, so it can affect people at a very young age. Now, obviously, that looks like it peaks and then goes away, but that's unfortunately because we don't all live to 100. If we did, we would see that, in fact, the incidence keeps on climbing. And roughly, if you're an Irish male, you have about a 1 in 300 chance of getting this condition, so it's not that infrequent. These are studies from Ireland and Italy. So the number of people in the UK who have this condition at any point in time is about 5,000, we think, roughly, about 300,000 people worldwide, so it's not uncommon. The incidence is much higher, but people do not live very long with this condition, so the prevalence is lower. What do we hear from our patients when they come to see us? Well, firstly, they say that they have developed a disability. That's how it presents. It gives the problem from the outset. It may be weakness of grip, it may be tripping on the pavement, it may be slurred speech if it affects the throat muscles first. But the really difficult thing about this condition is it progresses. It progresses relentlessly, and people are then unable to use their arms properly to hold objects, to cut their food, to feed themselves, to toilet themselves, to wash themselves. And eventually, if it affects the throat, they're unable to speak properly or speak at all. They have difficulty swallowing their own saliva, and eventually they have difficulty breathing, and that is what limits life. Now, people for a long time said, well, the good thing about this disease, if there's one good thing, is that it doesn't affect the mind. It does actually affect the mind to a very subtle degree in about 30 to 50% of people, and that is something I can talk about a bit later. So the really challenging part of this condition is it's not relapsing and remitting, and there is no really effective treatment that can arrest it or even give you some relief from it. It's progressive, and the amount of disability you get increases. What you know is that what you've learned to come to terms with and accommodate this month will change, and that you'll face new disabilities and new challenges in the months to come. And although this is a relatively uncommon condition, it is the most common reason that people seek euthanasia. I won't go into the rights and wrongs of that debate. It's a completely interesting and separate debate, but it does tell you the personal challenge that people who have this condition face, that it is the most common reason. What's the biological anatomical basis for this? There are a whole series of neurons that can be affected, but the principal neurons that are affected are those that control movement, which is why it's called motor neuron disease. And there's a group of neurons that control the planning of movement in the frontal lobes. But then the execution of movement is elicited by the upper motor neuron and the lower motor neuron. And we see different deficits based on whether it's predominantly an upper motor neuron problem or a lower motor neuron problem. So the lower motor neuron problem causes major weakness. The muscles become very thin and they fasciculate, little flickering muscles that we see. Whereas the upper motor neuron does two things. It activates the lower motor neuron, but it also holds it in a state of readiness and damps down its automatic excitability. And so when the upper motor neuron is failing to work, we see stiffness and spasticity, exaggerated reflexes, and what we call the extensor part of response, the test that neurologists love to do where they scratch the bottom of your foot. In this instance, the toe goes up. In most adults, the toe would go down. So these are the challenges that people face. Interestingly, sensation is rarely affected, and eye movements are retained. So some of my patients who are no longer able to communicate with their voice actually have an eye gaze mechanism whereby they can select letters and communicate through the computer by picking out letters and making sentences. Bladder and bowel function is usually spared in this context. Well, what tests do? We don't have a test for this disease. There is not a single blood electrical scan test that we can do that says, aha, this is motor neuron disease, without a question of a doubt. What we do is we do a variety of tests that exclude other conditions. This is a rather cynical picture, but in fact what he's doing is he's stimulating electrically the muscle, the frontalis muscle, and getting contraction. But we do test the nerves in the body to see whether there's a neuropathy that might be mimicking motor neuron disease. We also scan particularly the spine. This is, you can't really see the MRI terribly well here. You can have problems in which there's compression of the spinal cord due to bony overgrowth or discs, and compression of the nerve roots, and that can look like motor neuron disease. So we always try and image the spinal cord and the brain in that respect. And we take blood tests because there are some autoimmune conditions that can attack the motor nerves in a very select fashion. And they are important to exclude because it is actually a treatable condition. We can give drugs that suppress the immune reaction and reverse the motor deficits. So those are very important. Okay, so the diagnosis can be difficult to make. It often takes 12 to 18 months to make. And of course, survival is only 3 years from symptom onset on average. So people can really face a very short time from getting the diagnosis to death, and that's a really challenging thing. Well, what can we offer in terms of treatment? There is one drug that works for motor neuron disease, but it has a modest effect. Now, most of you will not have seen the results of drug trials, and I will just talk you through this. The number of patients included was just under 1,000. And these are the people that were taking placebo. This is a survival curve. This is a probability of survival. So everyone at the start of the trial was alive, fortunately. And by 18 months, the vast majority of people weren't. And those people taking placebo, fewer of them were alive at 18 months, and every point along the scale than those who were taking riluzole. So riluzole was shown to be effective. And that drug became licensed in the UK and in most parts of the world. And has been validated by the National Institute of Clinical Excellence, NICE, as being an effective and cost-effective therapy for this disease. We looked at our patient population to see what we were doing was making a difference, and we looked at our dietary regime, our ventilatory support, our general impact of our care, and we were a bit disappointed to see that the only thing that had really made a difference in terms of our survival curves from our clinic population was riluzole. So we really do believe it does work, but if you have a 3-month increase over an 18-month period in a drug trial, that isn't really a very substantial change, and we need to do better than that. What else do we have? We have something called non-invasive ventilation. When you're no longer able to get enough oxygen in at night and release your carbon dioxide, we can give positive pressure ventilation to help with the respiratory muscles while you're asleep, and it really does make a very big difference. This is the overall survival curve. This is people who are on NIV in the blue line. These are people who are on, don't have NIV, who've elected not to have it, and they have a shorter survival. If you look at those people who've got limb onset, which is the vast majority of patients, they do much better. People with bulbar onset don't tend to do much better with this non-invasive ventilation. It doesn't just increase the length of your life, but also improves the quality of life. If you are retaining carbon dioxide and not getting enough oxygen, you wake frequently at night. And the one thing to make anybody in this room mad is to be woken every few hours by breathlessness. In the morning, people they retain carbon dioxide and they feel drunk and drowsy, and they often fall asleep during the day. Our patients go on this, they get a good night's sleep, they feel refreshed in the morning, and they don't tend to fall asleep during the day. So it is a really important an effective way of improving length of life, but also quality of life. So what's going on in the brain that causes MND? This is not somebody with MND. This is actually one of my colleagues, Martin Turner, but it is painted by somebody with MND. And I recommend that you Google this chap, Patrick the Optimist. As it happens, Martin is also an optimist, and he's got a quote here which talks about making one small step and work on motor neuron disease being similar to the Apollo mission. And yet my other colleague, also a great inventor, is Amaral Chalabi, and this is Patrick's portrait of him. I'm still waiting for my turn to have my portrait. Patrick wants to paint 100 portraits before he dies from motor neuron disease, and he's a really remarkable chap. So I do recommend his website. Anyway, so what I'm going to talk about is what's actually happening in the brain and the nervous system that leads to motor neuron disease. So this is a rather gorgeous graphic of the brain. What we see in our patients is that these upper motor nerve cells that are sitting in the motor cortex and the precentral gyrus, particularly layer 5, they degenerate. And they are the ones that are providing the input down to the lower motor neurons. And we've talked about the kind of symptoms and signs that they generate. And it's this population of motor nerve cells that actually sit in the spinal cord that make the connection out to the muscles that are lost as well. So it's a degenerative process that usually starts at one part of the body and then spreads to others. There are new theories about why that might be, and that the proteins that are accumulating perhaps being transmitted across synapses, and if you like, infecting local neurons, and this process progressing. I'm not sure that we've got convincing evidence of that, but what happens in the end is that almost all the motor neurons are affected, and people develop paralysis throughout their body. Well, what do we actually see if we look inside the cells? I'm not sure you can see this terribly well, but this is tissue in the spinal cord. These are motor neurons, and there's a brown thread-like globular structure that you can see within these cells, I hope. And this protein was identified in 2006 as being TDP43, the tau DNA binding protein, and its molecular weight is 43 kilodaltons. This was a huge advance for us. Before, we knew there were proteins that were accumulating, but we didn't know what they were. 95% of all people with motor neuron disease have this particular protein, TDP43, accumulating. And what's really significant is this protein normally sits in the nucleus. It's involved in regulating gene editing and transcription. And here it is, it's left the nucleus and it's aggregating in the cytoplasm. So it could be toxic because it's not in the right place doing the right thing, or it could be toxic because it's doing the wrong thing in the wrong place in the cytoplasm. So the other exciting thing for us was that this is not just a protein that accumulates in motor neuron disease, it also accumulates in a condition called frontotemporal dementia. Previously, these things were thought to be very separate. A dementia versus motor neuron disease. People with motor neuron disease didn't have major cognitive problems. People with frontotemporal dementia didn't have motor problems specifically. But in fact, this protein is found in about 60% of patients with frontotemporal dementia. And what we've subsequently identified is that there are families, in fact, that have dementia and motor neuron disease running through the same family. And just recently, we've identified a gene that accounts for both of these. Anyway, what is TDP43? It's a crucial protein that is involved in stitching together genes before they go and make proteins. I'll talk about that in a wee while. But it's a very, very sticky protein. It needs to be kept in the right place doing the right thing. Otherwise, it can aggregate. Now, this is what's called a Western blot. Now, it sounds very complicated, but it's very simple. What they've done here is they've taken the brain of people, in this case with frontotemporal dementia, they've ground it up, they've dissolved it in a series of strong detergents. So they start off with weak detergents here, and 1, 2, and 3. By the time we get 4, we're in a urea fraction. It's a very powerful detergent. And what you can see is rather than being soluble and just existing in this very light detergent, we're seeing a lot of TDP43 in the most powerful detergent that we know, 8 molar urea. Not only that, but there's fragments here, there's a fragment there, and it's stretched all the way up there, suggesting this is a very large, sticky protein which is accumulating in these brains. So it's not just present, it's stuck together in these aggregates, and that is part of the mechanism in which its toxicity is seen. This is not unique. Motor neuron disease, frontotemporal dementia, they're all part of a family of degenerative diseases of late onset that relate to protein accumulation. In the case of Alzheimer's disease, we see two proteins. One is beta amyloid accumulating outside the cell, so-called amyloid plaques. We also see these tangles of tau, the microtubule-associated protein tau, which forms inside the neurons, very similar to the kind of shape of inclusions we see with motor neuron disease. And in Parkinson's disease, in fact, we see these Lewy bodies in the cell, and they're made up of a different protein called alpha-synuclein. So there's a whole family of conditions. I've just mentioned 2 here, but there are about 15 in which different proteins are accumulating. These proteins become sticky, they go in the wrong places, and they start to cause disease. Now I'm going to talk about my favorite subject, which is genetics. That's meant to be a cartoon of DNA. And in fact, these are quotes that I made about 15 years ago, before we actually had much in the way of genetic evidence. But my feeling was, at the time, that it was going to provide the strongest clues as to the causes of motor neuron disease. And I say causes because although we call it one disorder, in fact, it's got multiple causes. And not only do we discover why it's happening, but we can use these gene defects to model the disease and discover how it's happening. Only when we actually understand the mechanisms of disease can we really make smart therapeutic targets and try and treat this effectively. So a bit of biology. Some of you are probably very experienced biologists, but not everybody in the audience is. I brought my children along. They don't know much about it. So I did promise my children that I would describe some of these things. I'm not sure whether you can see it terribly well because the lights are so bright. But this is meant to be a cell, and there's a nucleus. And that, I think even at the back you can see there's a chromosome there. And basically, chromosome is like a ball of wool, very, very tightly bound wool with DNA in it. And this is DNA. the genes, and they get read by polymerases, and they make mRNA. This is messenger RNA that's going to go on and tell the cell which amino acids to put together to make the protein. When we have a mutation, possibly just a single base change in the DNA, it leads to a mutation within the mRNA, and that gets turned into a mutation within the protein itself. And those proteins no longer behave the way they should, and they can become sticky, such as the case of TDP-43, and the other protein I'm going to talk to you about is FUS. So it's these proteins that are abnormal in this particular condition, motor neuron disease and frontotemporal dementia. And interestingly, these two genes, these two proteins, are actually involved in stitching together the mRNA when it's been read from the DNA. So that shows you how important they are. Well, what are we doing in the way of technology? The genes that we've been working on, we've been using this machine. This is actually a picture from my own laboratory. And it's what we call Sanger sequencing, named after Sanger. And basically, it's reading every single base singly in a tiny fragment. We can read about 500 base pairs at a time. It might take us a week to do that sort of experiment. And indeed, the first draft of the human genome was published in 2000. It took 15 years. It was just one person's genome and cost about £200 million. Things are different now. With these sorts of machines, we can do that in about a week for about £2,000. As you can see, it's a pretty big difference in terms of this new technology, and it's called sequencing by synthesis. Again, if the lights were low, you could see tiny little colored dots. And what we have in this machine is a very powerful microscope that looks at the way DNA is being made and looks at each base and records that. So we can do it about 1,000 times faster, about 100,000 times cheaper, and I'm confident that we will identify all the major genes that are responsible, certainly for familial motor neuron disease, and a large number that will be responsible for sporadic disease as well. So this is the graph of gene discovery. I came into the field around here when we only had the gene called SOD1, superoxide dismutase, and I used to give talks as I went through this decade saying SOD all so far, and that was true. But then, as you can see, it's almost an exponential increase in the number of genes. Not all of these are strongly associated with ALS, but some of them are. And the two genes that I'm going to talk about today are FUS and TDP-43. So it's pretty exciting times. This is how it looks in terms of our patient population. The vast majority of people have got no family history of motor neuron disease at all, 95%. But we do find gene defects. This is the new one just published about 3 or 4 months ago called C9orf72. It's chromosome 9 open reading frame number 72. And we find mutations in our patients of about 4% overall. In some populations, it's as high as 10 or 20%, particularly in Northern Europe. Some people have a family history of motor neuron disease, about 5%. And in those individuals, we find very frequently mutations. Now, more than 50% of people, we can actually detect a mutation in. The most common one is now C9orf72. In the UK, it's about 24%. But I recently screened a sample set from Belgium, and it was 86% of their undiagnosed genetic cases of familial disease. And in Scandinavia, Finland, Sweden, Norway, it's also very high, parts of Germany. The other previous gene that we had for all those years, SOL1, accounts for about 20%, and these two new genes, although they're relatively rare, they have given us insight into the disease mechanism. And you have to remember that although all these genes are implicated, it's this particular protein, TDP-43, which is present and accumulates in 95% of people. So it means that these other genes that are responsible for this condition are somehow mostly feeding into the TDP-43 pathway. So, how does this help us? Well, firstly, when people come with a family history of motor neuron disease, we can identify the gene defect. We can tell them why this has happened to them. We can also give them advice about other members of the family, so people who may want to be tested for the gene defect to see whether they carry the same gene defect. Obviously, it's a very, very challenging question to ask yourself to find out whether you carry such a fatal gene defect. But I do a lot of counselling with families in that regard. Everybody knows about genetics now, and even people who've got no family history at all realize that there are genetic risks, and so there are people that want to know whether they carry a gene defect. I think increasingly we're going to see people asking, even if they've got no family history, whether they carry any of these gene defects. And I think now with the C9orf72, you can make more of a case to do it. Lastly, we can make a difference for the next generation. Obviously, for a number of years, we've been able to screen prenatally for SOD1 in particular, and now we have other genes to do that. But not everybody could face the termination of a pregnancy, and that's fully understandable. So now what we offer is something called pre-implantation genetic diagnosis, which is where you do in vitro fertilization, basically fertilize the eggs in the laboratory. When they reach the 10-cell stage, we can take a cell out. If you could see that, that's what it would show. And we can genotype that cell. We can see whether that particular embryo carries the gene defect. And those embryos that don't carry the gene defect, we can put back in the womb, and hopefully they'll go on to deliver a happy, healthy baby. And we are now doing this for SOB1 in this country. So that, I think, is an important step forward. So although the population effect of familial motor neuron disease is small, and this is an important area for those families that are affected, the biological impact of these gene discoveries has been really huge. And I can talk to you about the number of times these papers have been cited and the kind of impact they've made, but it's a bit like a stone being thrown into a very still pool. The ripples are going out through the research community, and it really is changing all that we do. I'm going to talk a lot more about that in a minute. So this is the first protein, TDP43, this sort of curly-looking beast here. When the protein was identified in motor neuron disease patients, we immediately thought this was a good candidate gene. We screened 156 different families, and we found one mutation in one individual, this chap here, and I find variants all the time, and most of them are not significant. It took me a year to track down in different laboratories around the world these samples here, and I showed that the other person who was affected also carried the mutation, which I've signified by M, whereas the others had the wild type, the healthy copy of the disease, and they didn't get the disease. I then got contacted by this chap here, and he invited me to a family wedding. Not something happens very often to a researcher, but his family worked all over the world in the military service, and they were coming together for a wedding of this woman here. And his wife was not so pleased, but I turned up on the morning of the wedding with a lot of tubes and needles, and we bled most of the family. And these two brothers who were affected, who were at the wedding, we showed they carried the mutation, and all the other siblings didn't carry the mutation. And then lastly, this family from Australia turned out to have the mutation as well. Now I could see that the mutant variant was segregating with disease. So basically, those people that had disease also had this. This made me think it was more likely to be real. We also identified in patients who had no family history at all 2 neighboring mutations at position 331. This is 377. This is 294. Again, giving a bit more genetic evidence. But it wasn't until we actually did some experiments in tissues and cells that we saw real evidence for disease. So one of the experiments we do is to take an egg and look at the developing embryo. I don't know whether you can see that there at all. But one of my colleagues, Vinita Tripathi, can cannulate the central canal of the spinal cord in this egg, which is only a few days post-fertilization. And when she's done that, she can inject DNA and drive that into one side of the spinal cord, and you can see the cells that are starting to express the construct. And what we see here, wild type, and this is the mutant, we can see that SOB1 in this case is toxic. So we knew we had a model that worked. We did this now for TDP43. We injected the healthy copy. We didn't see cell death, but when we injected the mutant copy, we saw cell death, recognized by these black dots. Also, the whole creature was small and shrunken. In fact, doesn't live more than a day or two. So there was developmental delay and death in these that carried the mutation. And we were very pleased to have that published in Science in 2008. Subsequently, I've got colleagues that are working in the zebrafish. This is Corinne Howitt's work, modeling the disease effect in zebrafish. Frank Hirth is doing this in flies. And with Don Cleveland and I are generating mouse models with these gene constructs. I'm not going to go into that work in great detail. There's a lot to be said about that. What we are doing, however, is immediately getting on with generating cellular models to look for these high molecular weight forms of TDP43 under unstressed conditions, and under stress conditions we can actually see the aggregates forming in the cells, and we can use these to screen for compounds. And I'm at the moment trying to work with a pharma company to set up a platform whereby we can do high throughput screening of their compound library to look to see which drugs might be able to inhibit the aggregation of these proteins. So now I'm going to talk about stem cells. This is a bit of a funny story. I've been in this field probably about 6 or 7 years, and there have been some highs and lows. So I thought I'd take you through the highs and the lows of that. But we are making progress, and we've got some very new data which we're very proud of. So the first paper that really took my attention was published in 2003 by Tom Jesselstav, Heinrich Victorly. And what they did was they took these embryonic stem cells. Now, these are stem cells that are taken from the very earliest embryo, and they can turn into any cell in your body. You can't open a newspaper without hearing about stem cells. Well, these are the guys that do it. And what they did is they used two chemicals, retinoic acid and sonic hedgehog, which is a growth factor. It's really called sonic hedgehog. Somebody named it that. But what they did was using these chemical signals, which they'd identified in the spinal cord of patients, they could turn them into motor neurons. When they put these motor neurons into the spinal cord of a chick, they went to the right place where motor neurons live. They put their processes out in the right part of the nerve roots, and they made contact with muscles. Subsequently, they then showed that these mouse motor neurons were activating the chick muscle and were fully integrated. Now, there's a saying that we have where I come from, that if it quacks and it's got feathers, it's a duck. This is a motor neuron by all measures. So this is taking a completely undifferentiated cell and turning it into a functioning motor neuron in an animal. Quite a remarkable experiment. Anyway, so we thought we'd like to do this. The big idea was that we would make motor neurons in a dish from our patients. Easy? Well, no, it isn't. But we have achieved it just recently. The first idea we came up with was that we would use cloning technology. This is somatic cell nuclear transfer. You would have heard of it. The idea is you take a donor egg cell, you take its nucleus out, you put in fact, a whole cell, not just the nucleus. You then stimulate it to reprogram it, to make it think that it's an embryonic stem cell, starting to form an embryo. We take out the inner cell mass, we grow these pluripotent stem cells, we drive them to become motor neurons, we discover the drugs that cure motor neuron disease. Sounded fantastic. As like most of these things, it doesn't always work out like that. Anyway, the man that approached me to work on this was Ian Wilmot, who will be well known to you, and this is his friend Dolly the sheep. And we were going to use that technology, and we actually got a license to use human eggs to do this. Around that time, this man, Woo-Suk Hwang, claimed that he managed to do this successfully, use nuclear transfer to make an embryonic stem cell line from a human individual. So we flew out to South Korea on a couple of occasions. It was an extraordinary experience, which I could talk to you about ad nauseum in the bar. He did actually manage to clone this dog, which is why I've given this picture. And he's a lovely man, very sincere scientist. But of course, his work was discredited. Somebody in his lab was making up the results, and he and his team were horribly humiliated. He nearly went to jail for this. Around this time, in Shanghai, Hu Zhengsheng said that she could achieve this not using human cells, eggs, but using rabbit and cow eggs. And so when we had this disastrous encounter with Woo-Suk Hwang, we held a press conference, and I made the fatal comment, which was, of course, we can now use rabbit and cow eggs. And it's interesting to say this was warmly received by the press, who came up with headlines like Frankenbunny and Mutant. And you may remember the furore that happened with the whole animal-human hybrid embryo debate. There was very nearly a law passed that would ban this work altogether, and we spent probably about 9 months trying to persuade the politicians that that wasn't a good idea, and the public that we weren't cruel and devious scientists trying to create some sort of mutant, but that we're actually trying to work on an important disease that would help people. In the end, we didn't need to, because new technology came along. Anyway, at the time, there were some quite funny pictures that appeared on the internet about what an animal-human hybrid might look like. I thought I'd share these with you. Obviously, if you perform in front of a mouse, it can clap, that's quite good. You can look like your dog. I don't know why anyone wants to look like a cow, my personal favorite. And here's one we made earlier. Anyway, we were rescued from those sorts of disastrous and difficult experiments by an extraordinary man called Shinya Yamanaka. And he's actually an orthopedic surgeon. He wanted to make cartilage for people that were having problems with their joints. And he developed this technique whereby he could put in what we call early stem cell genes. So we thought we'd adapt this to make motor neurons from our patients. So basically, we take a skin cell from a person who has a gene defect which is responsible for their motor neuron disease. We put these early stem cell genes in, they then become these induced pluripotential stem cells, and they could be turned into making cartilage. Well, we don't want to make cartilage because we're interested in motor neurons. They can make gut. We're not interested in making gut because our problem is with motor neurons, but they do make brain tissues. And in the end, we were able to make motor neurons. Again, you can't see it in this light, but I promise you there are motor neurons there. So the idea here is not to make motor neurons to replace the motor neurons in this patient, but to actually be able to look at these cells and study the disease mechanism in a more normal fashion. We can't look inside the spinal cord and brain of our patients, but we can study the cells that come from our patients that have this in a physiological level. And when I was writing the grants to this, it was all quite science fiction, and we didn't know that we could make it. But, in fact, we have got some evidence that this is really a promising technique. So these are the cells we made from our patient cell line that has the M337V mutant, the one from that big family that I showed you. And these really are fantastic motor neurons. They make the right chemicals, cholinesthyl transferase. They are islet positive, HB9 positive, which is a marker for motor neurons. They are electrically active in the same way that motor neurons are. This is a human cell derived from a skin cell in a dish that is behaving like a motor neuron. Really quite remarkable. When we looked at these cells that had the mutation and those that didn't, we just followed them over time. This is one work done with Steve Finkbeiner in the States, and we saw that those cells that carried the mutant gene, they weren't treated any differently from those that didn't, but they died more quickly than those that just were from a healthy control. So there was something about having this mutant TDP43 protein that meant cells in culture did not survive as long. These are motor neurons. We used a HB9 reporter. So they die spontaneously. So there's something happening which is actually very much like what we see. And what we have seen, I haven't got the data here to show you, but we see the same aggregation of TDP43. It goes outside the nucleus, it forms these high mononucleate species, and it fragments. So we are looking at the disease in a dish in a way that we could only imagine that we could do from brain tissue previously, and that's very exciting. Not only we can start to see the molecular basis for why it's going to the wrong place and doing the wrong things, but we can also start to screen compounds that might reverse that process, which is very exciting. So the second gene I want to talk to you about is fusion sarcoma. This is actually the protein here, otherwise known as FUS, and this is the family that helped us discover this gene, and it's a very, very sad story. I met this lady when she was alive, and she'd seen all 5 of her children die of motor neuron disease in their 30s. And she lived long enough to see 5 of her grandchildren die of this disease as well. They survive on average 13 months from their first symptom. Yeah, usually they're dead within 6 months of diagnosis. And the weakness in this family always started in their arms. And the tragedy of this family is really very hard to fathom. But it did allow us to identify a region on chromosome 16 which carried a defective gene. We didn't know which one. In fact, there are 400 genes in here, and we were sequencing them very, very arduously. But we were working very closely with some excellent colleagues in the States, in particular Bob Brown from Boston. And one of the genes in there happens to be fusion sarcoma. And together, we identified mutations in this particular gene. And it happens to be, if you like, a cousin of TDP43. So we were very, very excited. It's another RNA processing gene. So these are the families that we saw it in, lots of different families that carried this gene mutation. These are the different mutations. And this is the position along the gene, and these are the amino acids that get added. And this is the very end of the protein. So you can see it's the very tail of this protein which is abnormal. And again, we were very fortunate to have that published in Science. So when we start playing around with these genes, we start putting them in cells, and this is the healthy copy of that gene FUS, and it goes into the nucleus, which is where it should be. That's the little cartoon of it. And we've tagged it with this green fluorescent protein, which you can see there. But when we put in the mutant that one of our patients had, instead of being in the nucleus, on top of the blue, it's outside the nucleus in the cytoplasm, so it's going to the wrong place. We think it's interfering with something called a nuclear localizing signal. When we chop that bit off, essentially a truncation mutation, it sticks outside in the cytoplasm as well, again suggesting that whatever's in there that's being disrupted by this mutation is responsible for taking it into the nucleus. And when we took the bit we chopped off, this NLS, put it back onto the mutant protein, it goes back into the nucleus, essentially rescuing the phenotype. So we now know that, in fact, the mutation is acting by taking it outside the nucleus and putting this protein into the cytoplasm in a very similar way to TDP43, although the mutations in TDP43 aren't in the same nuclear localizing signal. So it's a very interesting situation. We also showed these very large inclusions of this protein FUS in dead and dying motor neurons in our patients. So we had the aggregates that were present. This is, again, a very, very sticky protein. What we have done, this is one mouse experiment I will show you, is to overexpress the wild-type version of FUS, which is also toxic. Not just the mutants are toxic. And I'm not sure what you can see here, but here's a healthy mouse here. Here's a litamate, which has got too much of this FUS protein expressed as a transgene. It is smaller it doesn't move as well. Legs don't splay out in the normal fashion when you lift them up by the tail. And this is when they're put on what's called a rotor rod. It's basically a small rotating tumbler, and most mice are able to stay on this and happily trot around on this little rotating rod. But those mice that carry this extra copy of the FUS gene fall off very quickly, and indeed they die, almost all of them, by 120 days. So there's a very, very aggressive disease process when this is expressed in them. So how does this work? I'm going to give a quick summary of the biology that we reviewed. There's TDP-43, there's FUS. What they do in their normal function is that they stitch together the genes. So the whole gene is read across the DNA, but only parts of it, they're called exons, are put together, and these proteins are involved in the splicing machinery that do that. They also traffic genes around the place. The mRNA, they take it out to the places where it needs to be delivered to be translated into protein. But what we see in disease is that they mislocalize. They go outside the nucleus where they should be doing most of their function, and they start to aggregate in an unprotected fashion. Then they need to be degraded by the proteasome, but there's something wrong with this process, or degraded by the autophagy process, but there's a failure of that. So anything that increases the tendency for it to sit out in the cytoplasm, or there's a decrease in the clearance by these two mechanisms, will lead to the aggregation of these proteins, and that's what we believe to be the toxic mechanism behind this. And it's interesting that they're both RNA processing proteins, so we've done a lot of work on that. Anyway, in 2006, where were we? For frontotemporal dementia, we had MAP-tau. We had nothing else. We didn't know anything else in terms of pathology. And for ALS, we had SOLB1. We didn't know anything else. But in 2012, we now have TDP-43, which accounts for a large proportion of patients with frontotemporal dementia, and we have 95% of people we know have got this protein TDP-43 deposited, and a small number have FUS. Also, interestingly, FUS appears in frontotemporal dementia as well. Well, where are we with the genes? In FTD, we have this very brand new gene which affects both C9orf72, and many other genes account for it too. But you can see that we still have a long way to go in terms of identifying the genes that feed into the deposition of these proteins. So I'm not out of a job just yet, and we don't have all the answers. But the idea is when you have a new gene discovery, it's very exciting because we can immediately start putting into cells and discover why the gene mutation might be misbehaving, causing toxicity. We can then model the disease in very simple organisms like the fruit fly, the zebrafish, the mouse, but we also have a very important collection of brain tissues, the human tissue, which essentially is the perfect model. We are very grateful for our patients and the families for donating tissues over time so that we are able to validate the things we're seeing in these very simple animal models. But that's not all. What's really important is we want to discover drugs that are really effective. And having these cell lines helps us screen hundreds of thousands of compounds and take the very best of those to our fruit flies and zebrafish and the mouse to see whether we can inhibit or reverse the disease process. So I'm going to just show you my team. Whoop. This is my PA, Sophie Morris, who's not here tonight, and Vicky Strelchik, who's our lab manager. These are the gene hunters. Bradley Smith in particular, who's been with me a very long time. He was my PhD student in Caroline Vance. Bradley has done a lot of work on C9orf72, and Caroline has done the work on FUS in particular. I have a lot of people who modelling a variety of cell lines, and Emma Daniel did the work on the TDP-43 cell lines that you see. You can see Do Young is staring adoringly at her husband, Youngbok, and we're very pleased to have them both working in the lab. We also work on the brain tissues and the mouse tissues, and these are the people who work on the mice. So it's actually a white blood cell, and bouncing along the top of it are little crystallized structures. And this was a disease that was discovered when I was a medical student, and it killed people within 3 years. I'm not sure if anybody's in any doubt as to what this disease was. This is HIV. It was a virus. It was from a family of viruses we'd never seen before. We had no treatment for any viruses, and yet within 6 years there was an effective treatment brought to our patients, which has transformed the lives of people, certainly in the Western world, in terms of their future. And now people diagnosed at present may have a normal life expectancy. So there's nothing about motor neuron disease that makes me think that it's going to be incurable. A lot of the clues will come from genetics, but we really need to work this into cellular animal models and undertake complex drug screening before we can really make a difference. So the challenge to me and my colleagues is that we need to do smart experiments, that we need to have funding for this, and that we will make progress in terms of preventing the aggregation of these proteins. So these are my collaborators from around the world. I won't go through them all, but I will mention the work is supported by a large number of very important organizations, the Motor Neuron Disease Association, who funded me for nearly 15 years. The Wellcome Trust, indeed. The Wellcome Trust actually brought me over to Cambridge in the first instance. I've had a lot of support from the Medical Research Council and the Heaton Ellis Trust. Now, this is a fantastic group of friends of a family who are affected by this condition, and they have raised an extraordinary amount of money and helped my laboratory really significantly. I've also had funding from the American ALS Association that I'm grateful for, and people have donated to our local charity, the Psychiatry Research Trust. Most recently, I've got a studentship from the Wellcome Trust and some support from the Alzheimer's Research UK. So that is not the last slide, because there's some information. I would recommend this will be available on the website, so you can go to these organizations, the Motor Neuron Disease Association, the American ALS Association, and the Heaton Ellis Trust, that have fantastic information for patients about the kind of work we do and the kind of problems that people have. And then there are stories, the patient stories. And this health talk online, which is basically gives an account of people's experiences. And there's one particular clip from YouTube that I have got that I want to show you, but it is quite powerful. I'm going to show it after we've had questions. So if I could take some questions now. If there are no questions, that's fine too. And then I'll show that video clip for the last 8 minutes.
Well, thank you very much indeed, Christopher, for this very, very illuminating talk. Professor Shaw has agreed for about 5 minutes talk, and if you could avoid the technical questions, please, from our general ones. I think one I must say right at the beginning is anyone who sits around there and saying, why aren't they doing something about it, which you hear all the time, one realizes, of course, listening to this, that everyone is doing something about it, but everything is stepwise, by tiny step after tiny step. And this kind of research, and not only in this disease, but in many other diseases or afflictions, is expensive, very expensive indeed, both in money for running things and, of course, in the time and energies by various scientists. And I think you've given us a tremendous insight into what a clinical scientist in a major institution is doing these days. But questions, please. Straight in front of you, yes, please. Thank you very much.
95% of the cases are sporadic, yet it's a genetically caused disease. Does that mean that there's been a recent mutation to account for those 95%?
Yeah, this is a really interesting and important question. And I think I actually have got the answer. We've just got it in the last few months. What we've done, the most common gene in sporadic cases is actually this new gene, C9orf72. And we've mapped all the variation around there, and we can show that it probably happened from one single founder event, which we've estimated to be about 6,300 years ago. It's not perfect estimation, but it's roughly this date. So it's a single event, but the reason we see it in sporadic disease is that not everybody who carries the gene gets the disease. And we discovered it because we had these enormous families with 18 children, and the descendants of those had 10 to 12 children themselves. And we realized that this was a familial disease because a few of these people were having it. So it's what we call low penetrance. There are a lot of carriers who don't actually manifest disease, and therefore it'll appear to be sporadic. So I think the distinction between familial and sporadic disease is blurred. Even SOD1, there are more people with SOD1 mutations who've got no family history of the disease as who actually have a family history. So some of them are low penetrant as well, but it's a very good question.
Two in front, Lauren.
In the green jacket.
Here, yes.
Do you think induced pluripotent stem cells hold any kind of innovation for the future? Do you think they could sidestep the ethical issue of using embryonic stem cells and provide a better way of looking at curing diseases?
Yeah, absolutely. I mean, I started off in this with more hope than belief that they would really be informative. The problem is, your cells are still only a few weeks old in a dish, and we're studying a disease that affects people in their 50s and 60s. But what was really extraordinary was we saw the pathological changes that we saw in our patient's brain in these cells in a dish. And I think the reason is that growing cells in a dish is still a very stressful and an abnormal situation. And we can enhance that by giving them various chemicals and compounds, but also we can try and inhibit those processes. We hope that what we're studying in the laboratory in the dish is actually relevant to human physiology. There's a big jump there, but I think it really is showing us some very powerful insights. We've already identified 2 novel pathways from these cells that we think might be important in our patients, and they are potentially tractable pathways that we can treat with compounds. So there's a whole area of science that will come from this, not just in motor neuron disease, but in a lot of other disorders as well.
Thank you. Thank you very much. Could I ask what hope there might be for sufferers of Parkinson's disease from your area of research?
Right, so Parkinson's disease is a different set of proteins that are accumulating. But you notice that in Alzheimer's, Parkinson's, and motor neuron disease, these are all proteins that are accumulating abnormally. So we have a very sophisticated way of dealing with proteins. There's a housekeeping process. I talked about the proteasome and the autophagosome pathways. And clearly there's a failure of clearing of proteins, which is common to all these conditions. And something that I've been encouraging the geneticists who work on these huge data sets to do is to try and mix their data together to see whether there are pathways that are revealed. And I think that they're likely to be in terms of protein turnover and clearance. We have these cells for life. You know, they survive 50, 60, 70, 80, 90 years. So they have to have a fantastic system whereby they are recycling their proteins and clearing their house and making sure that everything is functioning. And eventually, with age, that fails. And I think we're seeing these conditions that are going to have many parallels. There's nothing specifically about TDP-43 or FUS that relates to Parkinson's disease. We do not see those proteins accumulating. But I think some of the downstream mechanisms in terms of the way cells cope with proteins that are aggregating abnormally will be in common.
Right at the front, far side, Lauren. Thank you very much.
Yeah, well, have you ever tested, because you test mice with the motor neurons, possibly because they've got very similar DNA to us, have you ever tried going one step further and using chimps, possibly, or a closer primate?
So there are people who are doing that in other diseases. In fact, in the UK, it's very difficult to experiment in that way on primates. And actually, I would find it quite difficult myself from a philosophical point of view. I think primates are very close and sentient beings. I don't feel the same way about mice, to be honest. I'm not sure that I would want to experiment myself on apes or monkeys, but I know others do, and there are some important experiments. Certainly, there's a model of Huntington's disease that exists in a monkey, and there's a sheep with Huntington's disease, so there are bigger animals that are being worked at, but that's not the kind of work we do. Thank you.
And also, have you ever, would it be possible for motor neuron implants, even though you said before that you weren't doing that, but is it possible?
So there are people who are trying to look into this. They're putting stem cells in the hope of replacing motor neurons. My problem with that is that motor neurons are the biggest cell in the body. They have to grow a very long way to make contact with the cells they need to communicate with. When they do make contact to possibly 1,000 other cells, they have to have millisecond precision in terms of electrical and chemical communication. And I think that complexity is just too much of a challenge. And our patients, of course, live a very short time on the whole. So the chance of saving them by putting these cells back in, I think, is remote. Also, we know that it's a very toxic environment. If we take the spinal fluid from a person with motor neuron disease out, and we put it onto a spinal cord grown in the laboratory, it kills the motor neurons. So it's a toxic environment. If you put these cells into that toxic environment, it's very likely that they would develop the same sort of problems. And that has actually been shown in Parkinson's disease. In people who were grafted 10 years before with Parkinson's disease, the new embryo-derived cells developed Lewy bodies with protein inclusions. So I think it's a toxic environment, and it wouldn't be, in my mind, sensible to try and replace those cells.
I'm very conscious of the time, I'm afraid. We've now got a short video.
Very short.
And that will have to bring the proceedings to an end.
But I
was told that this was not appropriate before questions.
So I should explain. This is a television clip from an Australian television channel talking about this one woman's experience. It's based around a video diary that this young woman is giving for her children because she has motor neuron disease, a familial form of motor neuron disease.
Well, Sarah Turk is a devoted mother of 2 beautiful young children. She also suffers from motor neuron disease, and this devastating condition is slowly killing her. But in a gift of love that will help save lives, Sarah has recorded a video diary. We'll let her speak for herself.
I want you to give Mummy a big smile. Show me how big your smile can get. You used to call out in the middle of the night, Daddy, Daddy. And when Daddy'd get there, you go, take me to Mommy, take me to Mommy.
I hate to see you cry, lying there and that.
And Logie, as you would say, Mummy loves you to the moon and back, or around the world 13 times.
So turn off your tears and listen.
What have you got, Hensie?
Sarah is planning for the future, making memories for 4-year-old Logan and 2-year-old Hannah.
You got to make a wish and then blow it into the air. Make a wish.
I remember when Loki was born, and he was born with the red hair, and he was little chubba chubba. And we got him home and both Mummy and Daddy didn't know what to do because you were our first baby. Hensie's first words were singing words. He sang, oh, the hokey pokey, oh, the hokey pokey. So beautiful. It's hard when you're talking into a video to try and remember everything, but just a couple, you know, anything to hold on to, I think, is something good. So I don't want them to live in sadness, though, that they don't have me. But I just want them to recognise
their mum. It's all happened so quickly. Just 6 months ago, Sarah was in training to join the Air Force when she started losing strength in her legs. Tests confirmed it was motor neuron disease.
Yeah.
There are moments, maybe quite a few moments, where I get a bit sad, and it's mainly just sad. Yeah, but just smile all the time, so I don't know.
She's got a killer smile.
Yeah, not bad. That was one of the draw cards.
You're an idiot.
Jason has taken leave without pay from the Navy to care for Sarah. She can no longer walk and is having trouble breathing.
Logie, you know songs from movies. Like if we hear a song on the radio, you'll be able to pick which movie that is. I know that sounds like a funny talent, but it's very bizarre, but you're very clever at it. And Hensie, you just know too much. And you're a bit bossy. Honey, you tell Mummy and Daddy, don't get cranky at me, I'm cranky with you. Or if we say, go to your room, Hensie, and you say, I'm not going to my room, you go to your room. And you're only 2.
It's a disastrous disease. It's the cruelest disease that if you set out to design the cruelest disease you could think of, this is it.
Even when she tries to be cranky, she's still got a smile hidden under there. It doesn't take much for it to come out.
It's not fair, though, because he's cranky and then just stays cranky if I'm cranky. He just smiles and then I smile and then I'm not cranky anymore.
It's a typical summer scene in the suburbs, a family get-together. But this family is anything but typical because motor neuron disease is affecting everyone. It's in the genes.
In our immediate family, my grandmother died when she was 72. Mum was, 50-odd, and now Sarah, at, how 26. 26. It's just, it seems like it's getting younger and younger.
It was only in recent years they realised in their family motor neuron disease is hereditary. Everyone has a 50-50 chance of having the mutant gene.
So she's inherited from her mum this gene that predetermines that at some stage in her life her motor neurons are going to get sick and die and this disease will kill her.
And how many people in, say, the last 10 years or so have died?
It's been, it's probably been close to 10.
Just take a look at this family photo. Sarah's grandmother Joyce died in 2002 and her mum Pauline died the same year. Uncle Kenny passed away in 1995. Suzanne was diagnosed in 2001 and Gay was diagnosed in 2006. Kathy died in 2002 and she had 2 children who both died young, Ashley in 1996 and Stacey in 2006. And now there's Sarah.
You're allowed to be angry. You're allowed to be sad.
Sarah has recorded a separate video message for any relatives confronting the disease in future.
And hopefully in doing so, I can prevent other family members from living a life of worry. Because I worried. And then I get here and I get diagnosed with it.
Talking to her children was toughest of all. One day, Logan and Hannah will sit down to hear Mum's stories.
Logan and Hannah, I wish that, Mummy being sick was, all that it just finished here for you guys and Daddy. But the truth and the sadness is that it doesn't.
There's a 50% chance that either of Sarah's children will have inherited this gene.
So many things in life make people sad. But the sadness that I have is so deep. Auntie Naomi is in the midst of organising a charity for when you guys grow up, if you want to have kids. There'll be money there for you to go through IVF and have babies that don't have the gene.
It's perfectly feasible, and so it should be done. If it's a charity that can be set up to get rid of this gene abnormality that destroys lives and puts unbelievable pressure on family, then it has to be done.
Logan and Hannah, aka Logie and Hensie, I love you both. To the moon and back, or as Logie would say, around the world 13 times. I love you more than anything.
It's not fair, is it? It's very hard to watch. The very brave Sarah Turk, Ben Fordham reporting. And if you'd like to help, donations can be made to the Sarah Turk fundraiser, BSB 802-249, account number 2618-7905.
For more information, please visit www.gresham.ac.uk.